Hello, everyone. Welcome to 2022 WIU Graduate Research Conference. My name is Oluwapumi Ojo, a graduate student in the Department of Biological Sciences. I and my colleague will be presenting the proposal of our research titled Affinity Maturation of a Pancreatic Cancer Targeting Phage.
Good day, everyone. My name is Theodora Ide, a graduate student in the Department of Chemistry here at Western Illinois University. We both work under the mentorship of Dr. Mette Sondergaard, a professor of biochemistry in the Department of Chemistry here at Western Illinois University.
The pancreas is an essential organ in the body. It has both an exocrine and endocrine function, and it aids meal digestion and also regulates insulin production. Pancreatic cell has the highest mortality rate of 95% among all major cancers all over the world. This is linked to its low 5-year survival rate of less than 3%, and this is due to inadequate early detection method. The already known detection tools for pancreatic cancer, which include the computer topography scan, endoscopies, all these tools are only relevant at the advanced stage of the disease. It is therefore very imperative to develop early diagnostic tool so as to be able to initiate early therapy and to improve the survival rate of pancreatic cancer patients. The objective of our study is to develop an affinity maturation library of a pancreatic cancer targeting phage. A previous study which was conducted in our lab was able to use phage technology to identify MCA1 as a peptide that binds to pancreatic cancer cells, the MIAPACA2 cell lines. And it does this at a specific and high affinity with an EC50 value of 97 micromoles and high C50 value of 2.15 micromoles. MCA1 has 14 amino acids, and we hope with this study to be able to improve the binding affinity of MCA1 by developing a new phage display, which consists of phage that display scrambled peptides of the same amino acids that were found in the original MCA1 peptide. This peptide can then be used in another phage display selection to identify optimized peptides that target pancreatic cancer cells.
Now, I'm going to talk about how the library is going to be amplified to ensure minimal damage of substantially reducing its diversity. Amplification is accomplished by infecting fresh cells with a portion of the library. Now, 2 1-liter flask is going to be inoculated with 100 ml of terrific broth with an overnight culture of K91 glucan. This overnight culture is grown in NZY medium, which acts as a medium. NZY medium is shown here in figure 2, and I hope you can see my mouse. It's shown here in figure 2. Now, NZY medium acts as a medium, and is going to be inoculated with 100 microgram per ml of kanamycin, which is an antibiotic. This would shake in a lab shaker shown here in figure 3. And this process degenerates F. pili, which are flexible filaments elaborated in the F1 cells of E. coli. Now, the shaking is going to slow down for 5 minutes, and the shed F. pili is going to regenerate. 10 to the 12 power of physical particles of the library is going to be added to each flask. Each culture will be poured in a pre-warmed 3-liter Fernbach flask containing 1 ml of NZY, supplemented with 0.22 microgram per ml of tetracycline, another antibiotic. This is going to shake for 35 minutes at 37 degrees Celsius. 1 ml of 20 milligram per ml of tetracycline will be added to each flask to bring the concentration of tetracycline to a total volume of 80 microgram per ml. 70 microliter sample would be removed from each flask for the dilutions, and will continue to shake these flasks overnight vigorously. 200 microliters of serial dilutions will be spread on NZY plates containing the 2 antibiotics that we talked about, that is tetracycline and kanamycin. The colonies will be counted the next day. Virons will be purified from the colonies. In order to check the quality of the library, we would extract single-stranded DNA from a sample of the viron and sequence the degenerate insert region. The degenerate positions show up as obvious series of superimposed small peaks. Now, this new phage that we just got, the library we just got, is going to be utilized in another new phage display to find optimal peptides that target pancreatic cancer cells after it has been produced. Now, this is the overall bulk of the work because we're trying to figure out early detection for cancer cells, as my colleague said. In the future, the amino acid sequence with the highest binding affinity to MIAPACA2 cells can be used to construct a pancreatic cell early detection tool. Thank you for listening. And if you have any questions, do not hesitate to drop it in the discussion forum. I'll get back to you.